Okay, children, can you see me? One of you, Leila madam, will you please unmute yourself and tell me, can you see the screen?
Yes, I can see the screen. And your voice is
And
also clear.
okay, thank you so
You can
much. Now
start.
we'll start.
Yeah.
Yes, yes. Now we'll revise a little because whatever we are going to discuss this part right now is related to the previous part, and always once we recap, that's better. Okay, and I hope you have learned at home, you have studied, you have read whatever we have discussed. Please, as soon as whenever we discuss any topic, start reading, drawing whatever is told. Now today's part is regulation of gene expression. Now, last time we have learned the process of protein synthesis, which involves two steps. First is transcription, and the second is translation. So, last time we discussed translation, and previous to that, we discussed transcription, right? So, first is the transcription, and the second is the translation. Okay, related to that only, we are continuing with this part today now. Transcription, now in case of eukaryotes, these are all different levels, like transcription, where primary transcript is formed, which is considered to be immature or non-functional RNA. Then the processing is done on it, that means splicing, capping and telling. Then this mRNA is ready, it becomes functional and ready to move to the cytoplasm for the process of translation, the second part of protein synthesis, and then translation takes place. Okay, so in this way, these are the steps involved. So, and there is control of the genes for each type of processes. Each cell in the human contains all the genetic material for the growth and development of the body. Some of these genes will be needed to be expressed all the time, but there are some genes which are not required to express all the time, so they are either they are made on and off type of phenomena is there available. So some biochemical vital processes like respiration needs the functioning of gene continuously, they work, but there are some genes which are not required to be expressed all the time. In that case, switch on and switch off mechanism is done. Now, example, the best example I'll just tell you in case of this is Lac Operon. Let me tell you the concept of operon first. Operon is nothing but a group or cluster of genes that regulates the production of enzymes needed to degrade lactose in E. coli and some other enteric bacteria. Let it be forget about lac. Just let me tell you what is operon. Operon is a concept used to study the functioning of genes in coordination. There are different types of genes present on DNA, right? Some are structural and some are functional. So these structural and functional, these genes operate together. And so that concept is considered as operon. Operon is a concept where there is cluster of genes, or the genes are arranged in a sequence, specific sequence, and they work coordinately, and then the function is happening. Now, we are going to take one example of Lac Operon. Lac is the word which is the Latin word, means milk. Okay. Now here we are going to where the milk contains lactose, right? So that lac has come from the word lactose, and lac means milk. That is a Latin word. Now, how this is the concept used here in this example is when the E. coli, like bacteria, they are provided with the medium which contains lactose. And when it is not available, then what happens? Now, before we go in detail, just let me show you the structure of this. This is the double-stranded DNA you can see. Now, these are the genes. Now, try to understand. See, these are the names of the genes I'm showing here. This is the promoter end. This is the terminator end. And these are the LAC A, LAC Y, LAC Z. I'm showing from the right side. Just remember, this sequence is always in this way. I'll explain this part again, but just try to understand this diagram first. So LAC A, that is for this is the gene called LAC A, called Lac A, this is Lac Y, and this is Lac Z. These three are the structural genes. Okay, and immediately next to these structural genes, that means next to Z, Lac Z, operator gene is present, and next to operator gene, promoter gene is present. And all these activities are again regulated and controlled by Lac I, or this is also called as regulator gene, which is denoted by either capital R, or this can be considered as Lac I. Now, what is that Lac I? What is Z, Y, etc.? I will explain you now. But first, you try to understand this. This is the DNA. And last time, why I repeated and recap last times, because we have studied transcription and translation, and you know that what is required to start the transcription, that is DNA double stranded structure, which becomes unfolded by when the RNA polymerase gets attached to that part, and then unwinding of that DNA strand takes place, and one of the strands work as the parental strand for the synthesis of mRNA or whatever the different types of RNAs. Remember this point now. Now see what is Lac Operon. Lac Operon consists of 3 genes, each involved in the processing of the sugar lactose. Just now I showed you 3 genes, 3 structural genes, okay? Not only 3 genes, but these are only structural genes. One of them is the gene for the enzyme beta-galactosidase. And this enzyme hydrolyzes lactose into glucose and galactose. You must be knowing that lactose is disaccharide, and when it is digested, it gets converted into glucose and galactose. These are the monosaccharides. Now what happens? This is the example. In case of E. coli, lactose sugar is provided to the culture medium. Cell induces production of 3 enzymes necessary for the digestion of lactose. See, when the medium, nutrient medium, where these E. coli cells are grown, that nutrient medium is provided with lactose as its nutritional part, then this requires, or this induces the production of important enzymes which are necessary for the digestion of lactose. Now, what are these enzymes? Let us see first. First is beta-galactosidase, and its role is what? It digests lactose into galactose and glucose. As I told right now, that lactose is disaccharide, which is digested to, means it's converted into 2 monosaccharides, galactose and glucose. Second enzyme is beta-galactoside permease. Now see, the name itself suggests permease means it gives permission. What it permits? It permits lactose molecule to enter into the cell so that the further functioning will happen. Now the third important enzyme is trans-acetylase. That is also called as beta-galactoside acetyl transferase. The name also suggests. See, transferase means what? Something is transferred. What is transferred? That is a group. That is acetyl group. From acetyl group coenzyme A. That means this transfers an acetyl group from acetyl coenzyme to galactoside. So this acetyl group is transferred to galactoside. See, these are now these enzymes which are necessary for the digestion of lactose. Now, which are the genes responsible for these synthesis of enzymes that we will see? Now, these are the 3 structural genes I showed you in the diagram. That is Lac A, Lac Y, and Lac Z. This start from the rightmost part, that is beta-galactoside trans-acetylase. That means this is the Lac gene which encodes for this enzyme. Then Lac Y gene, it encodes for beta-galactoside permease. Okay. And then Lac Z, it encodes for beta-galactosidase. That means Lac Z. Now, if you remember the diagram, I'll show you the diagram again, but just remember, starting from the right, that is A, then Y, then Z. These are the 3 structural genes. Then immediately next to that, O, that is the operator, and then P, that is the promoter, and then R, that is called as the regulator gene. I showed in the diagram. I'll show you again in the next diagram. That is also called as the Lac I. I means that I represents either inducer, or it may show the work as inhibitor. That is also called as repressor. Now, synthesis of these 3 enzymes is controlled by segment
Yes,
of
one
DNA. Yes, ma'am.
Excuse me, Mrs. Joshi here. I want to read one important notice to the class A and B division. May I please for a minute?
Yes, please.
Thank you so much. Students, very good afternoon. This is a notice for you all for students of standard 12th A and B division who want to change their subject from biology to geography or from mathematics to geography are requested to send application with parent signature through WhatsApp, okay, to their class broadcast number. You all are aware which is the class broadcast number for you, okay? To that number you have to send the application. Please send the application latest by Thursday, that is tomorrow, 9th July 2020. Please note that the change of subject will not be entertained after that. Those who have already sent the application, they need not have to send it again. Okay, but if you wish to change today and tomorrow, 2 more days are given, but tomorrow will be the last day. Please note down, talk to your parents and accordingly send the application. After that, it will not, no change will be made in any subject. Okay, because the list will be finalized. So after that, there will be no change. So tomorrow is the last day for it. I hope everybody has noted it, and we have sent the SMS and messages also to your parents. So I hope you all are aware now. Okay, so this was the important thing about the change of subject. Thank you, madam. Thank you so much.
Yes. Okay. Now, children, listen to me now. See, synthesis of these three enzymes is controlled by a long segment of DNA known as operon. I showed you in the diagram. Okay. So it consists of operator site O and three structural genes at Z, Y, A. The action of structural genes is regulated by operator site with the help of a repressor protein. Now, what is that repressor protein? I will show you again in the diagram. Okay, it's produced by the action of gene I called as inhibitor, known as regulator gene. I showed you that this is called as regulator. The the where it is located, I show you in the diagram. Okay, when this protein is attached to the operator, blocks the switch is turned off and structural genes are not expressed. The gene expression depends on whether operator is switched on or switched off. That I will show you in the diagram. Okay. See now, this is the diagram. Just first try to understand the diagram again. Okay. This is the DNA double stranded structure. As you know, I told you when the RNA polymerase gets attached, the unwinding of the two strands takes place for the process for the synthesis of new strand of RNA, right? So RNA polymerase gets attached, and this is what all related to the process of protein synthesis. Try to understand. Now, see, this is this is represented as I here in this diagram. This is the regulator gene, which is represented as I. Okay, and this is the operator site. These three structural genes at Z, Y, and A. Okay, that means lac Z, lac Y, and lac A. And I told you what is the role of lac Z, which is responsible for the synthesis of beta galactosidase. Then this, what I said, is permease, right? And this is trans-acetylase transferase, right? So immediately next to these structural genes, the operator gene is present. Okay, what is the role of operator gene? Now let us see. When RNA polymerase is attached ready for the process of protein synthesis, that is the first step as transcription, then what happens here is the regulatory gene, which either works as inducer or works as inhibitor. Okay, then that means it is regulating the further processes. Now when lactose is absent, see, when lactose is absent in the medium, what happens? A repressor protein is synthesized. When lactose is not present in the medium, then what happens? Repressor protein is synthesized and it sits on the DNA in front of the O. That means it gets attached to the operator site. And so when this repressor is attached to the operator site, what will happen? It blocks the synthesis of RNA polymerase and the further process of mRNA synthesis doesn't take place. And though the RNA polymerase is attached, but when in case of the medium lactose is absent, it produces this I gene, regulatory gene, it produces inhibitor called repressor. and that protein is attached to the operator site, and that's why it blocks the further process. That means what? These three structural genes will not take place, will not work. They will not work because this operator site is blocked. As the RNA polymerase is not available, the further process of transcription will not take place, because RNA polymerase cannot bind here, as the repressor is produced by inhibitor, that is the regulator gene, and it binds to the operator. So it will not operate, it will not allow the operation further of the structural genes. Okay, so in case of lactose absent, when absence of lactose is there in the medium, no lactose is available, then repressor gene, it gets, that repressor protein is produced by the regulatory gene, and it blocks the operator site. So operator gene will not function, and the further process of protein synthesis, that is the first step, transcription, that means the RNA polymerase will not work for the synthesis of new strand. Transcription is stopped here. Now, if the operator is switched on, that means it was switched off. Now, when operator is switched on, that means what happens? The genes ZYA will be transcribed by RNA polymerase into a single mRNA. And before I show you the next diagram, just try to remember these two terminologies. Each structural gene is generally known as cistron and is transcribed, and the transcribed long mRNA covering various cistrons is called as polycistronic. Now, what we are showing in the diagram is that is the polycistronic. Many cistrons are present, many structural genes are present, right? Now, switching on and switching off of the operator is achieved by a protein called repressor. Just now I showed you that repressor. Now, what happens when lactose is present? See, again, this is a double-stranded structure of DNA, but this strand is single-stranded structure I have shown because this only is working as a parental strand, template strand. Now, what happens? This regulatory gene or I, what is the role here now? See, when this I is present, now means this I or regulatory gene, what it does when lactose is present, it produces a specific type of sugar. When small amount of lactose is present, allolactose, this type of sugar is formed. And this, because of this, what happens? This fits in to the operator, this sugar, that means this allolactose type of sugar, what it does, it binds to the repressor. You can see in the diagram, this is the repressor and this is the sugar. So it binds to the repressor in such a way that it has to change its shape. And when there is that change, there is change in the shape of the repressor's gene or the protein in such a way that it cannot bind to the operator. And as the repressor is not able to bound to the operator site, that RNA polymerase is ready to function and work, and so the further structural genes as Z, Y, A, they are operated. That means what? Because of the presence of lactose, which forms its another form that is called as allolactose, and that binds to the repressor protein in such a way that it cannot bind to the operator site. So this operator site is free, and then it promotes the synthesis of RNA polymerase, and it works. RNA polymerase works, functions, and so these structural genes, that means Z, Y, A, they function, and there is synthesis of new strand of RNA. Okay, now this is what about the Lac Operon or lactose metabolism. This is called as Lac Operon. Now this is the what in the tabular form I have just given you, shown you that what is the LAC-Z, what is the role of LAC-Z, that means beta galactosidase, and what is the function. Okay, means what it codes for, LAC-Y, what it codes for, and what is function, LAC-A, what it codes for, and what is the function of the enzyme. That is just shown in the form of tabular form. Now this is what operator LAC-O, promoter LAC-P, repressor, and LAC-I, that means regulator. Now, the next part of this, I hope you have understood this, but before that, I just want to show you the
expression of can be transcriptionally regulated.
You will see the video, which will be, see this. This is the Operon in eukaryotes as well as prokaryotes. Now we are talking about E. coli. This is Lac I, that is regulatory, promoter gene, operator, and the structural as ZYA. Okay?
Three genes clustered together chromosome are required.
They are called as structural genes. Immediately next to structural gene, operator gene. See the role of LAC-Z, beta galactosidase, synthesis, and lactose permease is synthesized by LAC-Y, and galactoside transacetylase by LAC-A gene. This is the promoter side. Yes, it transcribes. This is the polycystronic mRNA. It produces. This is the operator gene.
Unit consisting of Lac promoter, Lac operator, and the Lac Y gene called the Lac Operon.
The entire structure is called as Lac Operon. And this is the regulatory gene called as, yes, this is the mRNA. In absence of lactose, this repressor is produced. And this repressor joins to the operator. When it joins or binds to the Lac operator, this RNA polymerase will not recognize and it will remain there. It will not be able to get attached to the promoter. And so there will be no synthesis of the mRNA. Now when there is presence of lactose, what happens? Allolactose sugar is formed. It gets attached to the repressor and it changes its shape in such a way that it cannot bind to the operator side. It cannot bind to the operator side. But then what happens? Then promoter gene can function. Then RNA polymerase is able to get attached to the operator, and then the further process of transcription takes place. And this is what polycystronic mRNA is produced. Okay?
Lactose permease and galactoside protein.
Okay.
Therefore, the induction of the Lac Operon enables bacterium efficiently transport lactose into the.
So now we'll come to the next part. This is now the next part is genomics. Now in short, I'll tell you about the genomics. That is what? Genomics, this is a term which represents the total genetic constitution of an organism. That means it's a complete copy of genetic information, that is DNA, or one complete set of chromosomes. That means monoploid or haploid of an organism. Okay, and genomics means what? It is the study of genomes through analysis, sequencing, mapping of genes along the study of their functions. The sequencing of yeast, Drosophila, mouse genome was done in order to facilitate the comparative studies between the humans and other organisms commonly used for genetic studies in laboratory. Now, in brief, I'll just tell you about why it is required and how it is divided into two parts. It's a very short, small part of included in the syllabus. That can be structural genomics, where mapping, sequencing, and analysis of genome is done. And the next part is what? Functional genomics, which deals with the study of functions of all the gene sequences and their expression in organisms. For example, now, how these functions are useful, how it is useful in the day-to-day life, that is, it can be used in, that means its role I'm telling about, that means in agriculture, to develop transgenic crops like tomato, rice, cotton, etc. Then genetic markers can be developed, where its applications can be used in forensic science. Then it can lead to introduce new gene in microbes to produce enzymes, therapeutic proteins, and even biofuels. Means in human study also, this is very important, even in the welfare of the human beings, like biofuels and all, natural environment. So what is HGP? In short, I'll tell you about HGP. That is nothing but Human Genome Project. Human Genome Project, first of all, was that is nothing but the to determine the complete sequence of nucleotide base pairs that make up human DNA and all the genes it contains. It's considered to be the largest collaborative biological project, and the idea was picked up in 1984 by US government, and it was planned, started, and launched in 1990 and declared to be completed in 2003. It was expected that it will take 15 more than 15 years, but before that only it got over, was completed. The work was completed. Now, just in brief, I'll tell you about the participating country and funding agencies. Participating countries were basically US, then UK, Japan, France, Germany, Canada, China, and along with that, some outside the government sponsorship was also given by Lac Operon, and the main funding two agencies were there, that is the Department of Energy and National Institute of Health Science. That is, these were the two funding agents. And few important points I'll just give you in brief. That means it was launched in 90 by US Department of Energy and National Institute of Health, and it was 13-year long publicly funded project. And the main objective of this Human Genome Project was to determine the DNA sequence of entire euchromatic human genome. So, what are the goals we will discuss about the Human Genome Project? First is to identify and map all 20,000 to 25,000 genes approximately, that is considered to be in human DNA, from physical and functional standpoint, which are the physical involved in physical way, or which are functioning in the body, to determine the sequence of 3 billion chemical base pairs that make up the human DNA. See, that was the main important objectives are these, that the sequence of these base pairs was to be studied. Then next is to store this information, these all informations in database. Once the information is known, that is to be stored properly. Then to discover more efficient technologies for data analysis. Once the data is ready, it should be analyzed. So efficient technologies are to be used. Yes, ma'am. Okay. But I'm explaining. So it will be next a few slides. It will be clear. Yes. Okay. Yeah. Okay. Now, children, one of you, Aksha, just unmute yourself and tell me whether I'm audible and you can see. One of you, just unmute and tell me.
Yes, yes, ma'am, you're audible and
The message of Lady Madam that she's not able to see. So I think she's out of the session. So just, can you see now?
yes, ma'am.
And my voice is also audible?
Yes, ma'am.
Okay, good. Then I'll continue. So these are main goals of the HGP. Now, the next is what? Allow the private sectors to access the information and technologies that arise from this project. Means once it is allowed to use this information, then few more informations or the few more techniques can come forward for the further studies. So that's why. Then also to sequence the genome of other organisms. Why do I study the genome of sequence of other organisms? Because it's very important in medical research. Because so many organisms like mouse and Drosophila, their genetic structure is to some extent similar to the human beings. And the last and the most, now let us see the applications of HGP. The sequencing of the human genome holds benefits for many fields from molecular medicine to human evolution. Okay, means if you study at the molecular level, in case of human evolution, in case of medical field, it's very important. This sequence, for example, once you know which is the gene responsible for a particular disease, then that can be replaced by the functional gene. So helps in identifying disease causing gene. Then identification of mutations linked to different forms of cancer. Now this is also a very important thing. Now, the cancer, so many cancers are developed because of the mutations. So once we identify what is the reason behind the mutation, or if any gene is responsible to for the mutation, then that gene can be replaced by the normal genes. So this is very important. The sequence of DNA is stored in database available to anyone on the internet. Then this is also one of the applications can be considered. Then gene bank can be produced. So this sequence should be in the database known as gene bank. And then to allow for advances in agriculture through genetic modifications to yield healthier, more disease resistant crops. Now in detail, I will explain you in the next part. We are going to study this in case of biotechnology in detail. So right now, I will just tell you in brief that there are so many crops which are developed using this technology, where it is considered to be one of the applications. Just now I mentioned about the BT cotton or the rice. Such are the products of agriculture. Then it should be benefited to the advancement of to know the victim at that place, if any accidental place or any murder or whatever. Okay, now these are few important points which I just want to mention you in case of human genome. See, very
Hello, madam. Can you hear me? Hello, madam. Can you hear me? Kotwal, madam. Kotwal, madam. Can you hear me?
Lele madam, there looks like some problem. Maybe
Yes,
the
yes.
internet connectivity is gone or something.
Yeah, I
Uh,
just messaged to her. Yeah.
yeah, ask her to set send resolution at 720 pixel. That also matters.
Hmm. 12 ते लेफ्ट द मीटिंग आला आणि तेव्हापासून आवाज गेलाय तुझा आवाजच नाहीये इकडे मला स्वातीलाही येत नव्हता मला येत नव्हता आता स्क्रीन पण गेलाय माझ्या इथनं बघ या या बघ जरा नेट गेलाय का Hello, Kotwal madam, can you hear me?
Madam, pause recording for a minute so that.
Yeah.